Good morning, dear colleagues. Again, my name is Anton Chernopyatko, and I am a member of research group at the National Medical Research Center of Children Health. I represent a group of authors, and they commissioned me to speak. My presentation is dedicated to the effect of deuterium concentration on intracellular parameters on cortical neurons during glutamate excitotoxicity. Deuterium depleted water for our experiment was produced at the factory in the center of Russia, in Kotovsk city. We use low temperature vacuum rectification to get it. This purification that's down to 1 ppm for deuterium and down to 300 ppm for oxygen 18. Its factory was built especially to produce deuterium depleted water. And now we increase our capacity month by month. Yes. Many central nervous system disorders are associated with glutamate excitotoxicity. Glutamate is the main excitatory neurotransmitter. It's a part of normal physiological signaling system. Under normal condition, glutamate acts very briefly. Wow. Okay. Very briefly. It's shortly open NMDA receptor and let ions of calcium 2+ goes into the cytoplasm. Calcium activate dependent enzymes and activate of many proteins, both in cytosol and in organelles. And then removed by special transport system, which consume a lot of ATPs. It's important for the future. Yes, and however, when glutamate acts because of some disorders too long and with high concentration, it causes excessive calcium influx into the cytosol. It leads to the structure and functional damage. It's, in addition, excessive calcium entry induced mitochondrial dysfunction, which may play a decisive role in neuronal injury and death. Mitochondria are the main buffering systems that help prevent excessive calcium in cytoplasm. Mitochondria tries to protect neurons. But when calcium accumulates critically in the mitochondrial matrix, this buffering system falls. Because of dropping down potential of inner membrane of mitochondria, it is goes down production of ATP, and it's because of calcium charges 2+, and it leads to depolarization of membrane potential, which used to produce ATP, and with some delay, mitochondria gives up and open permeability transition pore, big hole. And its accumulated calcium goes back into the cytoplasm and started to kill the other organelles, and in the end, it leads to the death of neurons. This phenomenon is known as delayed calcium dysregulation. Glutamate excitotoxicity is not a separate disease by itself, but it is recognized as a fundamental mechanism of neuronal death in many disorders associated with disability and mortality. And you see it's stroke, brain injury, schizophrenia, depression and anxiety, Alzheimer, Parkinson, even obesity. This can affect every person in the world, so it's very important. And now let me remind our previous conference. We demonstrated significant positive effect of significant positive effect of deuterium depleted water consumption on the most serious diseases. And most of these diseases was involved in glutamate excitotoxicity list. And of course, it was a task why it is. And in the present work, we aimed to look more closely at the possible cellular mechanism. It's me. The aim of the study was to investigate how deuterium contact affects neuronal viability, calcium dysregulation, mitochondrial dysfunction, and intracellular NADH in primary red cortical neurons under toxic glutamate exposure. Primary neurological culture were obtained from the cerebral cortex of Wistar pups. Wistar pups according to standard protocol. Sorry. All animal experiments were performed in accordance with the relevant regulatory documents and recommendation of European Science Foundation. Cells viability were determined based on the MTT test. And cells viability under glutamate excitotoxicity were assessed biochemically 24 hours after the end of glutamate exposure. Optical density in MTT assay was measured using multimode plate reader. By the way, it's very good equipment in this Russian institute, and it's full of the. We wasn't able even to find such equipment in the United States. Strange. Yes. After incubation of cortical neurons with high dose of glutamate in the presence of the glycine and calcium, we found a marked decrease in the number of viable neurons. It's a normal. It's 59% left of viable neurons after glutamate exposure. But pre-incubation of neurons in deuterium depleted water, 5 ppm, increased the number of surviving cells by about 20%. It's a result. For us was strange, but deuterium enriched water at 300 ppm and 1000 ppm did not significantly affect neuronal viability. This is the direction and where we continue to research, and we a lot of argue about this result, but the result is like it is. No effect, it's also a result. For to measure calcium, NADH, and membrane potential, we use fluorescent microscopy. For calcium and mitochondrial potential, we apply some fluorescent dyes. And for NADH, we use autofluorescence. Here's a sample, what we can see in the microscope. First stage is it's a buffer, just incubating of cortical neurons with the deuterium depleted water. Then this is a sample for the calcium ions, staining of neurons with fluorescent dyes. Then the third stage is the most informative phase during glutamate or proteinophore application, FCCP. Now we use this photo, and computer counted how bright it is and what is the color is the difference. And for calibration purpose, we use iodomycin. It's strong antibiotic. Typical curves of dynamic changes is presented here, and I can show what we are measuring. We measure lag period, which associated with delayed calcium dysregulation. This is time of delay. Area under the curves in two stages, glutamate and calcium-free buffer. We calculate this. And percent of recovery after we wash up neurons in calcium-free buffer. And we see that changes in calcium and delta psi are more or less synchronous. Of course, here's a minus, because it should be a reflected curve for the membrane potential. But it's direct numbers from the computer. Yes, and this is one of our real experiments. It's representative curves of dynamic changes. We see that glutamate causes massive influx into calcium into the cell. It's a lot of calcium. Opposite, substantially less calcium entered into neuron pre-incubated with deuterium depleted water. You see the clear difference. And almost all neurons were able to return close to the baseline. It means they are staying alive. Because here it's not there. Where after calcium dysregulation, it means that generally it's a death of the neurons. We made detailed analysis of effect of deuterium depleted water. And we see that, area under the curve was significantly reduced in case of deuterium depleted water compared to control group. Proportion of cells developing delayed calcium dysregulation decreased from the 61 to 47%. It is recovery graphs. And very interesting that this is lag period increase almost twice. It means mitochondria protect themselves much more efficient and survive twice longer. Recovery of calcium homeostasis improved by approximately 10% in deuterium depleted condition. But again, there are not significantly changes in the mitochondrial membrane potential in this experiment series. But we continue our research. Yes, now we found valuable effect for NADH, and this is for artificial intellect mice picture. And NADH is the main electron donor for the respiratory chain. It's transfer electrons to the respiratory chain, and in the end, it's produce ATP for our life. It's very important, and because of over 70% of neurons NADH pool is located inside of mitochondria, we can consider it as a important indicator of mitochondrial functional ability. We measure in whole neurons, but 70% in mitochondria, we can use it as a parameter. Yes, and we found that difference between the group of 142 ppm and 5 ppm was almost 17%, which suggests that deuterium depletion helps preserve neuronal metabolic status under exotoxic condition. It was measured by autofluorescence on the ultraviolet range excitation, and it was recorded neurons back at the blue range. By the way, it's rather dangerous for our students was to look into microscope when it's ultraviolet, and they always trying to remove the glasses, protecting glasses. It was some fight to keep the eyes. And opposite, effect of deuterium enriched water on the calcium 2+ ions and membrane potential in cortical neurons and the glutamate excitotoxicity, we see significant difference. Proportion of cells with delayed calcium dysregulation increased by 10%. The lag period increased by 10%. Lag period became shorter, about 80 seconds, compared with control group. The fraction of cells able to restore basal calcium levels during the post-glutamate period decreased by 9%. Decreased in membrane potential under the influence of glutamate was more significant, approximately 1.5 times than in control. Its results. Again, nice picture from artificial intellect. It shows that deuterium depleted water protects neurons from glutamate-induced energetic collapse, while deuterium enriched water exacerbates mitochondrial depolarization and calcium accumulation. Now its conclusion. Deuterium depleted water, 5 ppm, protects primary cultured cortical neurons from the death under condition of glutamate excitotoxicity. Deuterium enriched water, 300 ppm and 1000 ppm, do not affect neuronal viability. The neuroprospective effect of deuterium depleted water, 5 ppm, is due to increase in the accumulation of the calcium as a cytosol. A decrease in the deuterium content in the incubation medium promotes higher cellular metabolism, as evidenced by the high NADH level in neurons. In contrast, deuterium-enriched water promotes intracellular accumulation of calcium 2+ ions. Deuterium-enriched water, 300 ppm, significantly increase the drop in the membrane potential under the influence of glutamate, indicating mitochondrial dysfunction. This is conclusion of our work, and we continue our work. It's some intermediate stage. It's my acknowledgement for the team. It is Zanda Bakaeva and ladies in their hands was a real work, and also several unnamed students who help us. And also I want to thank Tatiana Strikalova, who was in the very beginning of this work. She generate key ideas and bring materials, and she will present another work after my presentation. So thank you.